Hello and welcome everyone to today's live class session of the week 10 lectures on the course Experimental Biochemistry by Professor Shagota Dasgupta and Professor Shaumo De of IIT Kharagpur. I am Srijan Sen and today I will be taking your week 10 live question and answer session. So as I always go, I will be firstly discussing with the lecture slides very briefly and giving a few important informations for a few important points that will be relevant for solving the assignments, and then definitely I will be solving a few questions that will also help you in solving your assignments and taking up the further exams, the future exams that are coming up in this month or anytime in the near future. Okay, so I'm sharing my slides with you all. Kindly confirm, and then we can begin. Okay, right. Right. I guess my screen is visible to you all. So without delaying any further, let us start with today's class. So basically in today's class, we would be talking about the basics of recombinant DNA technology. So in the last class, or the last to last class, we were discussing about the purification of proteins, the extraction of genetic material from different organisms for the genetic engineering for recombinant DNA technology. So the applications, we talked about this specific term of recombinant DNA technology, or rDNA technology. So today here we will be discussing about the basics of this particular recombinant DNA technology. So let us go ahead. So before we directly jump into recombinant DNA technology, let us first talk about DNA. We all know about it, but still just a very brief idea on this particular thing. So DNA, as we all know, is the deoxyribonucleic acid. It's a very large molecule that is basically made up of nucleotides, where you have sugar moiety, that is the ribose 5 carbon chain sugar. You have a phosphate molecule and a nitrogenous base, and this combines to form the nucleotides. When you do not have the particular phosphate molecule, when you only have the sugar and the nitrogenous base, we call it a nucleoside. A nucleoside plus phosphate molecule is your nitrogenous base, where the nucleoside is a combination of the 5 carbon chain ribose sugar and nitrogenous base, which is basically includes your adenine, cytosine, guanine, and thymine. So from this you can get ATP, CTP, GTP, and TTP. So these nitrogenous bases are responsible for carrying the genetic information from one organism or from one generation to the other. So what is a gene? So a gene is a long stretch of DNA that codes for a particular type of a protein that has different functions in a particular organism. So DNA that is first transcribed to RNA, and you can particularly say mRNA, and then it is translated into a protein that has several applications in the human system or any organism. Okay, so the basic material is your DNA. It's the unit of heredity in a living organism, and this is the gene that is responsible for carrying the information from one generation over to the other. So all living organisms depend on these genes. So genes hold the information to build, maintain the organism's cells, and pass the genetic traits to their coming offsprings or the future generations. So basically, what are the components of a gene? So genes basically composes of exons and introns. So exons are the ones that are a set of coding regions, and introns are the ones that are the non-coding regions that are removed sequence before therefore are labeled split genes or splicing. So what basically happens over here, so you have a gene comprising of exons and introns. So introns are the ones that do not code for any proteins, and when by RNA splicing, the introns are deleted from the particular gene sequence and only the introns get attached, or we can call them the introns gets spliced. And therefore what happens is that only the exons are the ones that are responsible for translating, rather transcribing and then translating in a particular protein that carries out different functions in the human body or in any organism. So genome, so we have heard of gene. So genome, it is called a library. So the genetic complement of an organism, including all of its genes. So all the genes that are collected together to form the genetic complement of an organism is represented in its DNA, and this entire collection of genes in an organism represents the genome. Now, this genome, what happens that there is a process known as the gene expression by which the information from one gene is uh, carried in the form of functional proteins. Okay, so these genes are basically translating into different proteins to from a functional gene product. Now, the process of gene expression is used by all known life forms, like you have the eukaryotes, you have different prokaryotes, viruses, that to generate the macromolecular machinery of life. So a gene is expressed, so a gene expresses to give a particular protein, and that protein is responsible for the different functions. So what happens in the different steps of gene expression is, first you have transcription, where you have the synthesis of mRNA, mitochondrial RNA. Then there is the post-transcriptional processes, different post-transcriptional modifications that happen, including the RNA splicing, where the exons are spliced together, kicking out the introns or the non-coding regions in the gene. And these exons, after getting spliced together, are translated into first transcribed, so they are transcribed and translated into proteins. That is comes translation, and then you have the post-translational modifications in the proteins. So if you can see from the figure, what we see over here is that you have DNA, then we have the synthesis of mRNA in the nucleus. So the mRNA is then moves into the cytoplasm. You have the ribosome, through which the synthesis of translation of protein occurs. Then this is the amino acids. It takes the help of amino acids to make these polypeptide chains, which further goes and folds into different secondary structures. Then the secondary structures are clubbed together to give a tertiary structure and ultimately a functional protein. So what are the genetic changes that you basically observe? First one is an alteration in the segment of DNA. That can be a mutation, a single nucleotide mutation, polymorphism, that can disturb a gene's behavior and leads to a diseased condition. So these kinds of alterations may be your mutation, where you observe a small genetic change, that is, you can call it a genetic drift. So when the shift or the change in the genetic makeup of that particular organism is shifted by a very small change, say a particular nucleotide or 2 to 3 tandem nucleotides, okay, repeated sequences of nucleotides, you see this is a very small change. So we call that as a genetic drift, just a small drift that has occurred, and that occurs due to mutations. Another one is a large genetic change that occurs by recombination. So you see a large sequence of the gene is inbuilt into the organism, and that occurs by the process of recombination, where a large segment of gene is cross-linked or crossed with another gene sequence, and a portion, say, of a particular other organism, a foreign organism is in inserted into our gets inserted into our genome, and there we call it as the recombination, and then an entire genetic structure or the makeup gets disturbed, because of which we observe a genetic shift. So these are the very two important points: mutation and recombination. So mutation is the effect of small genetic change leading to genetic drift, and recombination is a large genetic change leading to genetic shift, and this smaller or larger alterations can disturb the gene's behavior, leading to the diseased condition. So what is basically a mutation? Small changes in the genes, in the genome, that can be caused by viruses. You have certain mutagenic chemicals, certain radiations, transposons, that actually alter the sequence of the particular DNA. Like you have, for wild type hemoglobin it's CTT, but for mutant hemoglobin you have CAT. So the mRNA that is transcribed is different from that of the wild type hemoglobin. So what does this do? So if you read in triplets, so we know that proteins codes in triplets of the DNA sequence. So CTT, where we have the mRNA of GAA, codes for glutamic acid. On the other hand, CAT, that's transcribed to GUA, is a valine. So a one nucleotide change is basically bringing up a different amino acid, and the difference in one particular amino acid is responsible for a diseased condition, where we see over here that the wild type hemoglobin is converted to a sickle cell hemoglobin, and that's cause leads to sickle cell anemia. On the other hand, if we look from the perspective of recombination, that is the exchange. So this is a very important term, exchange. So corresponding DNA segments are exchanged between the adjacent chromosomes during the special types of cell division that gives an entire genetic makeup of that particular organism. Like there is a recombination from our organism, say organism A, and you have another organism B. So these two organisms A and B have some sequences that are interchanged among them. So a portion of B is moved on to A, and a portion of A goes on to B. So there is a change of a particular gene, and that can occur by different methods. So recombination can happen by through many methods, but anyway, so a larger portion of the DNA segment is changed, or a different DNA segment is inserted into the particular organism, thereby disrupting the genetic makeup of that particular organism, which can lead to several kinds of disease or different abnormalities. Okay, now very importantly, we have come to the recombinant DNA or the rDNA technology. So what is recombinant DNA? So DNA molecules formed by laboratory methods of genetic recombination, like we have heard of molecular cloning, that brings together the genetic material from multiple sources, creating sequence that would otherwise not be in a particular genome. So we do that in labs, or these are the experimental methods, the molecular bio methods that we do, like the molecular cloning for inserting the genetic material from a different organism into a particular vector, and then inserting that into a particular organism to see how that particular um, protein is getting formed, or that particular gene is expressing. So recombinant DNA is possible because the DNA molecules from all organisms share the same chemical structure. They differ only in the nucleotide sequence within the identical overall structure. So recombinant DNA is a general name for a piece of DNA that has been created by the combination of at least two strands. Our DNA molecules are sometimes called the chimeric DNA because they can be made up of material from two different species, and they are known as the mythical chimera. So basically, So what we do in recombinant technology, we isolate and clone single copy of genes or DNA segments into indefinite number of copies that are all identical. Now these new combinations of genetic material or these rDNA molecules are introduced into the host cells where they propagate and multiply. Now the techniques or the methodology is defined as your recombinant DNA technology. So it's very clear from the statement. For this molecular cloning, there are different restriction enzymes that we used. Okay, so there are these restriction enzymes are your bacterial enzymes that cut long strands of nucleotides into smaller segments, into which we can also insert our different DNA strand or the clone that we offer or a desired DNA product. Now, it is very important to recognize specific sequences of nucleotides and cut the DNA at a specific site within the sequence. Now, in one chain, the sequence runs left to right, and on the complementary strand, the sequence runs from right to left. Okay, so if you observe for the restriction enzyme EcoR1, so it recognizes this particular site that is palindromic in nature, like it cuts at this point. So you get two fragments. So these are the sticky ends. You get the upper fragment of G, and then you have C. And then over here you have G, and it also cuts at the same position from the complementary strand as well. And you observe several sticky ends where you get two products with the sticky ends. The two ends are here, and then over here you insert your gene of interest, your desired clone, and then it gets ligated, and you transform it in a particular organism and see how it behaves. You can check the protein expression, the gene expression, and all. So it recognizes specific sequences of the nucleotides and cuts the DNA at specific sites. So these sites are palindromic in nature, and the sequence. So over here you see that the sequence runs from left to right, and from the other side you see it runs from right to left. So see, if you can see over here that restriction endonucleases cut DNA at specific sequences, they recognize palindromes, and you see that this is a palindromic sequence and it cuts over here. Okay. The other thing about these restriction enzymes is that the single chain tails of DNA are called sticky ends that are created on each of the DNA segments by the action of restriction enzymes, and sticky ends readily bind to complementary chains of DNA. So the pieces of DNA that have been cut with the same restriction enzyme can bind together to form a new sequence of nucleotides, and restriction enzymes can be used to isolate a specific gene. Speaking about the cloning vectors, now cloning vectors are DNA carriers used to clone a gene and transfer it from one organism to another. Now, once a gene has been isolated, the small piece of DNA can be placed into a cloning vector or can be introduced into an organism. Many bacteria contain a vector known as plasmid. So you see there are plasmid vectors, and these are the DNA fragments that are to be cloned. So first we amplify these particular products by a polymerase chain reaction, and what we do over here, we enzymatically insert the DNA into the plasmid vector. So how we do it, we cut the plasmid vectors at the molecular cloning sites. So molecular cloning sites are the recognition sites of multiple restriction enzymes. So there are different palindromic sequences present that act as a restriction site for multiple restriction endonucleases. And then what we do, we cut the vector, we digest the vector, as well as we digest the DNA fragments, and then we ligate it using the a ligase Okay, and what happens after only when these are forms a circular, so when it forms a complete plasmid, only then it can grow inside the E. coli cells, and then you have the particular resistance that acts as the selectable markers for only specifically growing those plasmids. Okay. So first what you do, you first do the molecular cloning where you are amplifying the, so this is the entire process of molecular cloning where you first amplify the DNA fragment that you want to insert in the particular plasmid, in the particular cloning vector. And then what you do is that you in a restriction digest this particular plasmid and as well as the DNA fragments, and you ligate it and finally transform it to see which of the cells have taken up your colonies. So what is a plasmid? It is a ring of DNA found in bacteria in addition to its main chromosome. So that's a different thing. So it has its own chromosomal DNA that, so it can be, so the plasmids can be used to transport genes from one organism to the other. So a bacteria, particular bacteria has its own genomic DNA that is important for the regulation of its own activity for the survival of that particular organism. So those are the things that is present. Apart from that, if you want to insert a plasmid for growing to check the gene expression, we can use certain kinds of bacteria and transform the plasmid into that bacteria, specifically E. coli and all, and then carry out different experiments. So what is happening over here? So the process of using a cloning vector. So plasmid is removed from a bacterial cell and then it is cut using the restriction enzyme, and the specific gene that is isolated from a different organism or from a different species also has the sticky end. So this plasmid has the recognition site. This particular sequence also has these recognition sites, and we first restriction digest it, so it will generate the similar kinds of sticky ends, and finally when we are ligating it using the DNA ligase, we get the complete plasmid. The mismatches won't get ligated, and only the matched sections will ligate, and then finally we can carry out the transformation into a particular organism. So you see the plasmid is returned to the bacterial cell where it replicates as the bacterial cell divides. This clones the donor gene. The bacteria containing the clones of the donor gene can be used to infect other organisms and transfer the gene to them. So here you have the plasmid vector, the DNA fragment to be cloned. You get, so you are inserting this DNA fragment into this particular plasmid. So this is the recombinant plasmid, and then you mix E. coli cells, E. coli competent cells, and grow it on different plates, different resistant plates. You have kanamycin, you have different antibiotic resistance plates. Okay, so sorry, you grow them in antibiotic plates, like you have ampicillin, kanamycin, chloramphenicol, etc. So only those, so these particular plasmid has is resistance for a particular kind of antibiotic, so that will help you to basically select which of the cells have grown. So it basically selects the cells that will grow, because only those plasmids that have that resistance for the particular antibiotic will only grow, others gets died, other dies. So see, the cells that do not take up the plasmid die in the ampicillin plates, but the other, so this plasmid is ampicillin resistant, it has the ampicillin resistant gene, and this is the cell that survives and the cell multiplies, and from there you can get the recombinant plasmid. Now this is where you clone. In some cases what happens, plasmids are used to clone specific genes so that the bacteria will produce a specific protein. So for example, we have the insulin, that is a protein that controls the sugar metabolism. Similar kinds of transfer of the insulin gene is done over here, where we have the human cDNA, and then you clone the insulin gene, and then followed by transfection, and you grow them, so transfer and cloning of the insulin gene. Okay. Now coming to the protocol of the recombinant DNA technology. So how are you going to obtain a recombinant DNA technology? So the DNA fragment containing the gene sequence to be cloned, that we call as the insert, is isolated. Then we cut the DNA using restriction enzymes. Then we join the DNA using the DNA ligase, followed by insertion of these DNA fragments into a host cell using a vector that carries the DNA molecule. Now, the recombinant DNA molecules are generated when the vector self-replicates in the host cell. Now, the transfer of the recombinant DNA molecules into an appropriate host cell, followed by selection of these host cells using a particular selectable marker that we usually use as the antibiotic resistance or antibiotics, followed by replication of these cells carrying the recombinant DNA molecules to get genetically identical cells or a particular clone, and then we can isolate the plasmids from those clones and get our rDNA plasmid. So to the isolation, the first step of making this recombinant DNA is to isolate the donor and the vector DNA. So this procedure is used for obtaining the vector DNA. That depends on the nature of the vector. The bacterial plasmids are commonly used vectors, and these plasmids are purified away from the bacterial genomic DNA. So there is a protocol for isolating the plasmids from the genomic DNA, such that there is no genomic or the chromosomal DNA contamination. That is the own DNA of that particular bacteria. Now, ultracentrifugation, this is also used as a protocol for extracting plasmid DNA. So the plasmid's DNA forms a distinct band after ultracentrifugation in a cesium chloride density gradient containing Ethereum bromide, and the plasmid band is collected by punching a hole in the plastic centrifuge tube. Now, what happens in alkaline lysis? Now, another protocol relies on the observation that at a specific alkaline pH, bacterial genomic DNA denatures, but the plasmids do not. So, furthermore, subsequent neutralization precipitates the genomic DNA, but plasmids stay in solution. Now, phages can be used as vectors for cloning DNA in bacterial systems. So phage DNA is isolated from a pure suspension of phages recovered from a phage lysate. So for cutting the DNA, we have the restriction enzymes. That there are different restriction enzymes. You have HIN3, EcoR1, then you have BAMH1, etc., so many, that will cut the circular DNA molecule bearing one target palindromic sequence, resulting in a linear molecule with single-stranded sticky or blunt ends, depending on the type of restriction enzymes that we are using. So I have mentioned this before, that you have the recognition sequence over here, and similarly the sticky ends are generated. Similar kinds of sticky ends are also generated in your insert. So the vector and the insert have similar kinds of sticky ends that will help to ligate those portions into the particular plasmid. Right. Now for the insertion process, we have to choose a gene cloning vector. So a vector is basically any DNA material which is capable of multiplying inside the host to which our gene of interest is integrated for cloning. Now in this process, restriction enzyme functions as scissors for cutting the DNA molecule. Now, ligase enzyme is the joining enzyme that joins the vector DNA with the gene of interest. This will produce the recombinant DNA. Now, for introducing vector DNA into the host cell, we have the plasmid vector. This vector is added to a flask containing a culture of your E. coli, and then you have to make these E. coli cells competent. So, calcium ions in the form of calcium chloride are added to the flask, followed by a brief heat shock. Now this allows the cell membrane to be more flexible, creating holes that appear on the cell membranes, making it permeable to DNA and allows the plasmid to enter. Followed by, what we do is that we plunge this cell into ice, into the ice condition or inside the ice, such that the holes or the created holes are blocked, or they come together, there are no more pores. So, and then the plasmid remains inside the cell, and then this can be grown. Now the phage vectors are introduced by infection of the bacterial lawn growing on an agar plate. Now the culture or the growth of virus is made more difficult than the culture of the bacteria or fungi by the fact that viruses will only grow. So how do we place the gene in the vector? So we have the plasmid DNA. Now the DNA molecules are basically strong and can be easily separated based on their size. So we cut them, we extract them, the bacterial cells are then broke open, and the chromosomal DNA is centrifuged down. Now this leaves only the plasmid DNA and the supernet. So if there are different solutions that we use, first we use an RNase A for the interference of any RNA to avoid any interference. Then we have a lysis buffer, which includes your alkaline conditions of NaOH and SDS that breaks open the cell, and the chromosomal DNA as well as the plasmid DNA comes out. Now we have the neutralization buffer that includes a potassium acetate solution that helps in the refolding of the plasmid DNA as well as the chromosomal DNA. But the chromosomal DNA, being very large in size, will take a lot of time. So before it can just regenerate into its original state, we go for a centrifuge. Now centrifugation. Now after the centrifugation, the chromosomal DNA remains in the pellet, whereas the plasmid DNA remains in the supernet. We extract the supernet and then bind them to different columns, silica columns, and then we extract the particular plasmid by uh, eluting it with an elution buffer of tris or even water. So restriction fragments from the donor DNA are mixed with the plasmid DNA and are joined by the sticky ends. The initial extraction is due to the hydrogen bonds, but the sugar phosphate backbone is then joined using the enzyme called the DNA ligase. So this is the another same thing that we discussed. So cloning into a plasmid, you have a gene for the antibiotic resistance, and you have the foreign DNA over here. It generates the sticky ends, and you just use the DNA ligase for joining these sticky ends, and then it is inserted into the bacterial system, followed by you get clones of bacteria when you grow them. Now it is plated on antibiotic plates, and only those bacteria that have taken up this particular plasmid or are resistant towards the particular antibiotic because of that resistant gene will only grow over here, and this can be used for DNA purification and extraction of plasmid. So this is all about the cloning protocol. Now there we have transformation. So transformation, there is bacterial transformation, like bacteria such as we have. Okay, just give me a second. Just give me a second. Okay, sorry for the interruption. Yeah. So yeah. So now transformation. So bacterial transformation. So bacteria such as E. coli can take up and express foreign DNA, usually in the form of plasmid, and then it is the gene cloning using the bacterial transformation to make lots of copies of the desired gene. So this is basically the same. So for transformation there is a particular protocol where we use the E. coli. For a bacterial host we use E. coli as a model organism because it is well studied. It does not have nuclear membranes. It has enzymes that is necessary for replication, grows rapidly, and is also inexpensive, and it's normally not pathogenic, and it's easy to work with and transform. So the second step of our bacterial transformation is to choose a plasmid to transform. We have the characteristics of a useful plasmid. The single recognition site, the plasmid only cuts in a particular place. So this ensures that the plasmid is reformed in the correct order. So the origin of replication allows the plasmid to replicate and make copies for the new cells. We have several marker genes that identifies the cells that have been transformed. So genes for the antibiotic resistance. So the bacteria is first plated on the media with an antibiotic, and only bacteria that have taken up the plasmids will only grow. So genes that expresses color. So the bacteria that have taken up the recombinant plasmid are of a different color than the bacteria that do not take up the non-recombinant vector. So finally, the bacterial cells are prepared for transformation by treating them with calcium chloride that softens the phospholipid bilayer and allows the plasmid to pass through the membrane. Also, electroporation is another technique by giving brief electric pulse to open up the pores of that particular cell membrane, or directly injecting the plasmid into the bacterial host. So the final step, so the other steps includes the plate transformation on appropriate media that contains the nutrient agar for bacteria and antibiotic to distinguish transformed bacteria from the non-transformed ones. And then these plates are incubated overnight at 37 degrees, and furthermore the next morning the plates are analyzed. So for this molecular cloning process, we need to carry out this polymerase chain reaction. So it was first described in 1971 by Klepp and Khorana and redescribed and successfully used in 1985. So this allows massive amplification of specific sequences that have defined endpoints, and this is a fast, powerful, adaptable, and simple process. And it has several applications. So to obtain the material for cloning and sequencing for in vitro studies, to verify the identity of engineered DNA constructs, to monitor the gene expression. Also, it is used to diagnose several genetic disorders, identify an individual, or reveal the presence of any microorganism. So now, why do we need this polymerase chain reaction for? So to know about this polymerase chain reaction, we have a template DNA that contains the target sequence. So the primers that are short oligonucleotides that defines the ends of the target sequence. So we have the thermostable DNA polymerase, so buffers and DNTPs, and then we have a thermal cycler. So we need these things for PCR. So the final finally what happens is that we carry out this PCR reaction, where the first step is denaturation, then primer extension, primer annealing, and finally primer extension. And this step is carried out for many cycles, like 30-35 cycles, and then the product is basically amplified. The sequence is basically amplified. So in this step there are new reactions that are taking place, and this is increasing in the number of two pieces from one product. So if you see that, so first you have one, then comes after one first reaction you have 2 to the power 1. So after 30 reactions you will get 2 to the power 30. So ultimately that is leading to this many number of gene sequences. That is an amplified product. Okay, and finally, okay, so that is the last one. So this PCR reaction is used for amplifying your target sequence, and then you do the cloning process. Okay, I guess that much is good enough about the recombinant DNA technology. If you have any questions, you can go ahead with asking me. Okay. Do you have any questions? Else we can start with the question with the previous year questions, the solving of the questions. Up till now, if you have any questions, you can go ahead. So, no questions? So, shall I start with the assignment? Good to go. Let us see. The first question is recombinant plasmids are added to a bacterial culture that is pre-treated with dash ions. Yes, so recombinant plasmids are added to a bacterial cell culture that is competent for taking up plasmids from the medium. So that culture is made competent by treating it with calcium chloride or the calcium ions. Okay, next one. To join two different DNA fragments, which of the following enzymes is used? Ligase, restriction, phosphatase, or polymerase? Yes, so you say 2A. That is absolutely the correct answer. Ligase enzymes. Third one, the process by which the plasmid is inserted um, into the host cell is known as transformation, ligation, annealing, gene cloning, by which the plasmid or the vector is inserted into the host cell. What do we call it? Just try, give it a try. What is it? The insertion of the plasmid or the vector into the host cell. Give it a try. Yeah, it's transformation. Very good. So that is, you're transferring. You are transporting the plasmid that is into that particular host cell such that it can grow, it can multiply, and you can get multiple clones of that particular plasmid. So transformation, that is correct. In PCR, magnesium cations are used. What is the main reason? So why are magnesium ions used? It gives extra stability to DNA molecules. It is a cofactor of the enzymes used in PCR reaction. It increases the cell penetrability, or none of the above. Yeah, very nice. So it is a cofactor of the enzymes used in the PCR reaction. Brilliant. So next up we have a match the following: gene of interest, plasmid, clone, vector. So try matching these components with the description. So gene of interest is a DNA segment that is to be inserted or deleted. That is correct. 1, 4. Then you have plasmid is a circular double-stranded DNA molecule that is distinct from the cell's chromosomal DNA. So 2 is 1. Clone is organisms that carry identical genes. 3 is 2, and 4 is vector that acts as a vehicle to carry the foreign genetic material into another cell. That is also correct. So A is the correct option. Question 6. What is the role of a selectable marker in cloning vector? Maintaining vectors in cell, selective survival of cells that have the vector, increasing cell growth, or expressing the gene in the cell. Yeah. So selective survival of the cells that have the vector. Also, the other option that is correct is A. That is maintaining the vector in the cells. That is also correct. So A and B both are the correct options over here. First, maintaining the vector, otherwise the vector will definitely die. And finally, it's the selective survival of the cells that have the vector. Okay. The next one. Multiple cloning sites, MCS, in a vector has. What does MCS have? Promoter site, DNA ligation sites, restriction sites, antibiotic resistant sites. Yes, so these multiple cloning sites have unique restriction sites for the recognition of different restriction enzymes and nucleases that will cut and generate different sticky or blunt ends for the molecular cloning. Fantastic. What will happen if the origin of replication is missing in a vector? Very nice. What will happen if the origin of replication misses in a vector? Vector will not be taken up by the cell. Vector will not contain the gene of interest. Vector will not survive in the cell. Vector will not replicate. No. Origin of replication is missing, so the vector will not replicate. Is the correct option. Why it will not take the gene of interest? It will definitely take the gene of interest because it is dependent on the restriction sites. It will also be taken up by the cell because the cell is competent enough to take up that particular vector. The cell will also survive in the cell because the antibiotic resistance gene is not missing. What is missing is the origin of replication that will prevent the vector from replicating. Okay, next one. Choose the correct sequence for the steps of PCR. So to renaturation, extension, hold, annealing, denaturation, annealing, extension, hold, annealing, denaturation, extension, hold, extension, denaturation, annealing, hold. Yes, option B is correct. Denaturation, annealing, extension, hold. That is the correct option. Great. Next one. The DNA sequence of the desired gene is given below. So you have a DNA sequence over here. Which of the following restriction enzymes will digest the above DNA fragment? So here you have to observe it and then you have to give an answer. So which of the following restriction enzyme will digest the above DNA fragment? So these are the things that are given over here, that what it recognizes, which enzyme recognizes what. So find it out in the DNA sequence and give an answer. So basically a restriction enzyme is one that cleaves DNA and at near specific recognition sites. So tell me which one is there. You want a bigger version of it? I can just increase the size if you want. Now see. Which concept is not clear? Tell me the question. Is it about this particular question? Okay. So what you have to find over here is you have to identify whether there is this particular sequence or not, TCT-AGA, or there is CAT-ATG or not, whether you have GTC-GAC or not, and whether you have GAA-TTC. So depending on which of the following site you find over here, you recognize what is present over here. So that will be the particular sequence that these restriction endonucleases will identify and cut over here. So the question says you observe this particular DNA sequence and find out which of the restriction enzyme will digest the DNA fragment. So that is, you have to find out the site. Find out the site for which the restriction enzymes will cut. Okay, so you find out which of the following sequence or sequences are here. So after TAT, you have GCC. So it's a continuation. So 2 more minutes for you to find out. No, no answer. Okay, you see AGC, CAT, ATG, and in D1 is the one that identifies it. So option B is the correct answer. Okay. Okay. Anyways, let's go to the next one. Which of the following statements is or are true? DNTP mix consists of mixture of restriction enzymes and primers. Exponential amplification of a gene can be done from very small amounts of DNA. Thermostable DNA polymerase is absolutely necessary for PCR. Antibiotic resistance is achieved through the use of selectable markers. So which of the following statements is or are true? So there will be many questions like this where you have to find out the correct statement. It will be an MCQ, but there were more than one option might be correct. So there will be many questions in the coming exam. So if you are giving the particular exam, be prepared for this kind of questions. So tell me the answer to this. Yeah, very good. So DNTP mix consists of DATP, CTP, DGTP, DTTP, all of these. Um, and these are monomers that make up the DNA molecules. It does not contain restriction enzymes and primer. All the other three options are correct. Great. Next one. Which of the following should be present in a vector? Cloning site, antibiotic resistance gene, origin of replication, all of the above. Tell me, which of the following should be present in a vector? Yes, all of the above: cloning site, resistance gene, and origin of replication. Next one. Which of the following codon is not a stop codon? So which is not a stop codon? TAA, TAG, TGA, or ATG? Okay. So you have heard of no idea. ATG is the start codon. It codes for methionine, and methionine is the start site of any protein synthesis. Okay, so ATG is a start codon. It's not a stop codon. The other codons are UAA, that is known as the ochre. You have the TAG, that is the amber, and you have the TGA, that is opal. So all the other three are stop codons. Only ATG is not a stop codon. It's a start codon, coding for methionine. The next one. The temperature cycles in a polymerase chain reaction are in the order. So what is the order of the temperature cycle in a polymerase chain reaction? Yes. Fantastic. Denaturation at 95, annealing at 60, and extension at 72 degrees. So that is the correct option. The last one, which of the following statements is true for the heat shock method? This method is used to kill bacterial growth. It increases the cell permeability of bacterial cells. In a process, the competent cells were incubated in 42 degrees for 45 minutes and the competent cells are heated at 45 degrees centigrade. So what of the following statement is true for the heat shock method? Fantastic. It increases the cell permeability of the bacterial cells. So option B is correct. So that's all for the question and answers. You have any queries on this or on any other class lectures? Okay then. Thank you. We will again meet in the next week. So thank you all. And these lectures are being recorded and will be uploaded in YouTube. So you can write down over there, or if you have my email ID, you can also send me a mail with the particular query. I will be very happy to answer them. So thank you.